help with answering some questions about the number 4 post-lab on enzymes. And students are a little bit confused about what the graphs should look like that we're making. And I think the confusion comes from, I think folks are used to thinking, we'll collect some data and then we'll make some graphs and then we're done. But we've already seen in Lab 3 that scientists will often make graphs and then use that data to do more work. And so we saw that in that spectroscopy lab, where we were working with cobalt chloride, and we said we got to do absorption spectrum to find out what's the best wavelength to study this molecule. Each molecule has a unique absorbance spectrum, and we need to learn about that to find out it. So we have absorbance here on the x-axis. This is the first graph we made, which is the cobalt chloride absorbance spectrum here, and this came up in class. So just to mention this, so the absorbance ranges from 0 to typically 1, and sometimes as high as 2 are accepted, but values, so between 1 and 2. And the reason is because the instruments start to become inaccurate above a wavelength of 1. That's fine. Just to tell you a little bit more about this, if somebody measured their absorbance, if a scientist measured their absorbance and the absorbance was too high to be likely to be accurate, what they do is they'll dilute the sample. And so you could just take your sample in a new test tube, and we didn't do this in class, but just to give you this general knowledge, we could add 9 parts of water and 1 part of sample. So we do it, that's a 10-fold dilution, a total of 10 parts, and 1 part of that is the sample. Another way to think about that is write this, if you look at this, the sample is 10% of the total value. Okay, so then we measure the absorbance again, and let's say we measure it now, and we got a value of the absorbance of 0.25. Okay, and and so the real absorbance must be 2.25 times that dilution factor, that 10-fold dilution factor, or the real absorbance of this would be 2.5. Too high to be measured, but it is fair to measure anything by diluting it. And that's because of this direct relationship between concentration and absorbance. If we dilute something 10-fold, the absorbance will dilute by 10 times. And so we didn't have that issue, but I just wanted to bring that concept up. So we're going to measure in the visible wavelength, and I like to, follow up. I wish we could follow up a lot on every lab, just to show you this, to get a feeling for this. So this is the visible light wavelength, and it ranges from about 400 nanometers to 700 nanometers in wavelength, and though that corresponds to the real colors from the rainbow. Roy G. Biv is the little mnemonic that people use to remember red, orange, yellow, green, blue, indigo, violet. Beyond violet, we have the UV light. Beyond red, we have the infrared light. And there are lots of biological molecules that absorb out here, proteins, DNA, RNA, and some that absorb out here, some other types of biological molecules. And so our instruments just use the visible wavelength. It's possible by an instrument that can also measure in the UV or infrared. And they just need a different light source and a different capacity of wavelengths to measure. So we measured, and we got data that looks like this. So it went up, and it basically went down. And that was our graph, and we didn't make that graph, but if we had, we would have found that the absorbance maximum was about 500. I think that's what students were using, and that's the best wavelength to study this molecule. So that was our first graph, or first data collection, and we really didn't need the graph. We were really focusing on just what is the maximum wavelength. Just to mention something, this is not the kind of data that we would try to fit in our course into a scatter plot and a standard and a trend line. This is the kind of data that we would fit as a line graph, and the data would go up and then back down. A line graph, the line is implying that if we went to this wavelength, the absorbance would be about there, that it would be in between these two. And so that's fairly accurate. The real data would have more of a curve to it, like a curve up and down, like that. So it's not perfectly accurate, but the best type of graph for this would be a line graph for that data. Again, we didn't really need to make it because we saw, yep, the highest absorbance was 500. That's what we're trying to get out of it. But in this case, this data could have been graphed, and we could have just said, look for the peak and use that. And that would have been using this as just a first step tool, because then we kept this wavelength and we took the rest of our measurements in that wavelength. And what we went on to do is we went on to measure the absorbance of these specific dilutions, and we got some data that looked about like that. And so again, absorbance and concentration in molarity on the x-axis. And here we don't want to make a line graph because in theory, because of that concept of that there's a direct relationship between absorbance and concentration. If we doubled the concentration, we double the absorbance. In theory, this should always be a perfectly straight line. And it should go through zero, and should be a perfectly straight line, right? Zero concentration, zero absorbance. So I just missed it in this hand-drawn graph, but yeah. So when we make the average line with our data, it's not going to be a perfectly on that because there's errors in our data. But we could draw it and say, here's the trend line, like that. And so the trend line is this average line, and that's what we really want to use as our value because it averages out errors. It does, it averages out some of the errors in our data. And it's not a perfect prediction, but that's what we want to do. And then we use that, and we called that the cobalt chloride. Let me just turn off this timer. Turn cobalt chloride standard curve. With this instrument we used and the molecules cobalt chloride, the absorbance should always fit on that curve, close to that curve. And so then we used it as a tool to measure the absorbance of 3 unknowns and later calculate their concentration. So the real data that we wanted out of all those experiments was just to be able to estimate the concentration of unknowns A, B, and C. And that was our final real data. So we run into the same kind of thing here in Lab 4, where we start out, we collect data, and we have to make a graph. Then we have to analyze that data, and then make a final graph. And so we have 3 different variables that we're trying to measure. So we're trying to measure the enzyme concentration. And we measured enzyme concentrations like this, rounded out to the nearest 8%, 17%, 33%. Okay. And so we collected data. And so we start out and we could make one graph with all these 3 on it. And this axis would be time, and this axis would be absorbance. And I'm just making up this data, but we could say with 8%, we started out at 0 time about here, and we collected data, and we got data here, and here, and here, and here. And then we collected data for the 17%, and let's say that we got data here, and here, and here, and here. Then let's say we collected data, and we got data for 33%, and we got data that went up like that. Okay, so the prediction is, and I mentioned in class, some of these start to tail off, but they should start out as linear. And why should they be linear? Why should there be a direct relationship? Because here's the enzymes, they're little molecules, and they're grabbing substrates and catalyzing reaction to make products. And what we're measuring is the products, and the enzyme should work at a steady rate. So that means it should make the same number of molecules every second. And the molecules are going to be what gets absorbed. And so if we double the time, we should double the absorbance. So there should be a direct relationship between production of product and absorbance. Okay, so what we want to do here is, this situation is there's going to be error in this measurement, but in theory, each of those would be in theory, a straight line. And so our best thing is to do a trend line here and get a one trend line for this, and then a trend line for this 17% data, and then a trend line for this 33% data, okay? And so that is measuring the rate of the enzyme reaction. The steeper the rate, the faster the enzyme is producing, and the faster enzyme produces product. And so here, I think the data I sketched out, it wouldn't be too surprising if the concentration increases, we would increase the rate. More enzyme produces more product because we have more enzyme molecules. And so that's kind of what our hunch prediction is. I don't know what your data looks like, but that's what our hunch prediction looks like. So what we're really interested in is the slope. And this first graph we make is just to create a graph to determine the slope. And so you should determine the slope of each reaction, and in Excel it'll give you this equation y equals mx plus b, and I think this b is just the slope of the graph. I'll double check that. I've just forgotten that at the moment, but yes. And that's the value we really want. Really want the slope. And if you haven't gotten that from an equation in Excel, you can calculate it, an estimate by hand. And the way you would do that is you would go to 2 points on this trend line, pick them, and just pick how much this changed over time. So how much absorbance increased per minute. And so you can either find this equation just by popping up on Excel next to the line, or you could just make a hand calculation of it based on that, because the rate is the increase in absorbance. So delta means change in absorbance over change in time. So pick a minute, 2 minutes, whatever makes sense to you in your graph, and determine that slope. And so we're going to get a value for that. The slope is going to be, a change is going to be like, 0.2 per minute is going to be the value. And that's going to be the slope, or 0.2. And so this could be 0.2, and maybe this one is 0.15, and maybe this one is 0.1, we'll say, for example. And that is the slope. And the slope is the rate of the enzyme activity, and that's what we're studying. So what the post-lab explains is you first have to make this graph for enzyme concentration, as well as enzyme temperature and enzyme pH. So we're going to have 3 graphs in the post-lab that look like that. They're going to have these trend lines fitted to the different data. And for the temps we measured, I had written it down. So room temperature, we could call that like 22 degrees C, and 40, 55, 70, and 95. Okay, pH, I think we have 3, 5, 7, 9. Okay, so for each of these, we'd have 4 data points, and 3 data points for this first one. And so we get 3 slopes, 4 slopes, 4 slopes. And what the book says, and I'll just draw this out for one, is that we actually want to take those slopes. It says make a table, but then it says graph them. And so this would just be a nice looking graph for the concentration. And what it would have is enzyme rate on the y-axis, and it would have then on the x-axis the 3 values that we measured. So the measurements that we took for the enzyme concentration were 8%. So this would be percent enzyme concentration, and 17 and 33. Okay, and so we just have 3 values for that. And so what I'm guessing is your values will look like this. That would be lower here, higher here, and higher here. And that's what we want to establish, is just what's the change of rate as we increase the concentration. And then you can draw a conclusion. You could say it looks like we have higher reaction rates at higher temperatures, or at higher concentrations. That's what I predict you'll get. Maybe your results are slightly different. And it's good to check in with other students' groups to see what data they're getting during the class, because then you can make that comparison. And you always want to analyze your data, but if your data is really messed up, you can say, well, I attempted to analyze my group's data, but it seems like there are problems with it. So to complete the assignment, I did another group's data, and you just tell me that that's what you're doing. That's fine. But also then try to explain what you think went wrong with your group's data, and that's scientific reasoning. Try to identify possible factors that might have caused trouble, in your data, and why you think there was a problem with your data, and why you think other groups' data might be more accurate. And it might be more accurate because you judge it to be more accurate because you checked with multiple groups. But anyway, that's that. And so let me get this sent out to you so you have hopefully time to absorb it. All right, great. Thank you.